Hello, my name is Charles. Welcome to the latest Oki Solo channel YouTube video. So today I actually wanted to take some time and talk about percula clownfish, because my pair of percula clownfish is almost reaching sexual maturity. They've been picking at rocks and stuff, and I'm really excited to see that behavior in my own home. But it's gotten me thinking about the species a lot, and so I just wanted to take some time and talk about them. So, do the magic of editing. Hopefully you were looking at my pair right now. So the larger one is Katie Heron, and the smaller one is Regina George. If you understand that reference, please chime off in the comments below. I would love to know who is a fan. Those names were picked because, so I originally got this pair almost exactly 2 years ago at the time of recording. And I'm going to give a big thank you to Sustainable Aquatics. I had made a request in the spring of 2023 for a pair of F1 percula clownfish from a pair that had a known collection location, and they very happily and excitedly provided me with a pair of F1 percula clownfish from a pair that was collected in Papua New Guinea. And so what's really interesting there is I'm fairly certain almost all of the collecting that has happened in Papua New Guinea has happened on the southern coast, which would be in like the Port Moresby, Fisherman Island region. And almost all the scientific literature comes from the northern coast, which specifically in the New Britain province, and a significant percentage of those papers are coming specifically from Kimbe Island. And so clownfish are really interesting to study in the wild because they have a very specific life history. So percula clownfish live in anemones, and in Kimbe Island, just as a quick aside, so almost all the statistics and numbers that I'm using here come from the paper First Genealogy for a Wild Marine Fish Population Reveals Multigenerational Philopatry from Salles et al. 2016. So if you're really interested in the topic, please check that out. I might pull some other fun facts from other papers, but the majority of what I'm talking about today is going to come specifically from this paper. So the life history of percula clownfish lend themselves really well to wild research, because since they're site attached, you can come back to the same site over multiple years and track how that pair of fish is doing, or who is replaced in that colony, XYZ. And so let's take another step back and let's talk about the life history of percula clownfish. So they are sequential hermaphrodites, and they are all born male, and they settle out on an anemone. They'll grow up for, well, females take over 2 years to reach sexual maturity, and males less time. And if they reach that point, then they're reproducing, if things are going well, like every 2 weeks, and each spawn can consist of a couple hundred fertilized embryos, and then those are like waved out into the ocean currents, and then those larvae are traveling on the ocean currents for about 2 weeks, and then they go through metamorphosis and they settle out, and they begin anew in an anemone. So this raises some really specific questions on how to conserve these fish, because so there's kind of this dichotomy between philopatry, which is where animals that are born in a specific region return to that region, and then there's dispersal, where fish are born in one region and then they travel to another in which they are reproducing in. So a great example of philopatry would be like salmon. They're coming back to the same stream, breeding with other fish that were born in that stream, XYZ. The big advantage of doing that is that it allows for local adaptation. The big downside of doing that is your risk of inbreeding goes up, up, up. And then a great example of the flip side would be something like gray wolves, where they're born in a specific pack, and then when they get big enough, they would disperse out and they would either join a different pack or start their own. Why does this matter with percula clownfish? Well, this matters because since they have that larval ocean current planktonic stage, knowing where larvae are coming from is really important. So let's say that percula clownfish are very dispersal. So let's string together, let's say 3 reefs along. Are the fish in this reef coming from, are the fish that are settling out in this reef spawn from fish in this reef, and then fish in this reef that are born here are settling out in this reef? Like, that means in order to conserve this reef, you need to conserve this reef. In order to conserve this reef, you need to conserve this reef. Or are the currents working that fish born at this reef are predominantly coming back to this reef? And yeah, it gets complicated really quickly. So Kimbe Island is a very specific study location and very interesting because it's largely consisted of 6 lagoons surrounding the island. And there are 2 main species, well, I shouldn't say main. There are 2 species of anemones that percula clownfish are found to host in them. I'm terrible at remembering the genus names of anemones. I only know the species names, so the genus name will flash across the screen the first time I say them. But so Gigantiae and Magnifica anemones. And Magnifica anemones are found further out, deeper water, and more exposed habitats, whereas, and these are generalities, whereas Gigantiae are found more lagoonish, shallower water, more sheltered habitats. So these are microhabitats in this island that is surrounded by 6 microhabitats and microclimates. That might be more accurate. And so in this study that I alluded to, what they did is over the course of 10 years, every 2 years they went in and sampled fish from each anemone, and which is so cool. But I think the paper said that they averaged 310 anemones in each collection. And over the course of 10 years, they sampled 2,972 fish. Now, that's intriguing. What's really cool is that, and I'm just going to start spitting some stats, is over the course of that, they managed to find some families, as many as 5 generations had passed in that 10 years, and they found 1,809 full siblings, 412 maternal half-siblings, and 248 paternal half-siblings. So this is my interpretation of that. That is intriguing to me. There's actually evidence that, higher up you get on the totem pole for a percula clownfish colony, which is a single anemone, the longer lived you are. So the non-breeding, non-dominant males have the highest mortality rate. And it seems to be, much like my pair, competition amongst each other forces them out, and they actually have a pretty fast turnover rate. Your life expectancy goes up the higher up you are on the totem pole. So dominant males have a higher life expectancy than non-dominant males, and dominant females have a longer life expectancy than dominant males, and vice, and so on. So having more maternal half-siblings would make sense. That means that the females are reproducing longer than the males are on average. But what's really intriguing is that most of the siblings that they found were full siblings, which means that for most pairs, for most of their lifespan in these reefs, during the course of the study, they had the same reproductive partner during that time span. There was also something else that really stuck out to me, is that traditionally speaking, the idea is that percula clownfish are terrible swimmers. They would never leave their anemone, and fish that do get nailed or eaten immediately by a predator. They actually found 2 examples of extra pair matings. And wow, that requires some determination. I think they said one of the extra pair matings was like 10 meters away, and then the other one was like 50 meters away. And that is quite the distance to travel. That is a level of determination that I'm not sure I will ever experience in my own life. That's insane to me. One of the things that was really interesting in this paper too was that there was evidence of certain individuals over-contributing. So that means that all the reproductive pairs at any given time were not contributing equally to the next generation in that population. And that raises the question is, are there fish that are genetically more competitive, or are fish in specific anemones performing better than other ones? Now, the evidence in this paper does not exclude the idea that there are fish that are more competitive, but within the data set they had, it seemed to me pretty substantiated that within the limits of their data, which anemone you were on in which regions mattered the most, and where that success happened changed on any given year. So one of the big trends was that Gigantea inhabiting clownfish contributed more to self-recruitment of that population at Kimbe Island. And that meant that the largest extended families on Kimbe Island were from fish that had multiple successive generations of settling out onto Gigantea anemones. Now, that doesn't mean that Magnifica inhabiting clownfish weren't reproducing as successfully, because that there's actually some evidence that since the Gigantea are closer inshore, they are more likely to self-recruit. And then Magnifica anemone fish are more likely to disperse their offspring into nearby islands. And so on any given year, the average self-recruitment of the population was something like 50 to 55%. And what was really cool about that is which lagoon of the 6 lagoons was contributing most in any given year did vary. And so that's one of the things that I think I was taking for granted, because we tend to think of currents as these like very consistent things, but that ignores that water is turbulent, and like, so currents on a large scale are pretty consistent, but flow isn't laminar and per se. And so there's minor variations in temperature and density, and that mildly changes the currents on any given year. And so like maybe one reef is contributing to that population the most in one year, but it might be beat out by another reef next year. That's intriguing to me. So let's go back to that self-recruitment statistic. Somewhere around 50%. That means around 50% of the fish on that island are not born on that island. And so that indicates, even as something with a short larval period as clownfish, like especially when you're comparing to like tangs or something like that, they're pretty interconnected. And so theoretically it might be possible for Kimbe Island to like sustain itself with only itself, but is that going to be good for the population? So one of the things that was really intriguing to me is inbreeding was actually really rare. I think they found one case of a full sibling pair and one case of a grandmother-grandson pair. And so this is like hundreds of pairs over multiple years. And so that just indicates to me that this is not a population that will handle inbreeding well if we're based off of past history. Generally speaking, that populations that are slowly selected inbreed will handle it better, whereas populations that don't experience inbreeding don't have any incentive to figure out how to handle it, so they don't. And also, like, this population was fairly diverse for its size. There was like 300 pairs at any given time if we're saying that each anemone had a pair. So what are the implications of this? I think the big one is that these populations of percula clownfish are probably way more interconnected than we thought possible. We don't know where this 50% of fish from other reefs are actually, like, we don't know which reefs those are coming from. And that also means that we don't know where a lot of the Kimbe reef clownfish are going. And so that means conserving these could be really complicated. Like, it's theoretically possible that Kimbe Island, if you completely isolated, might be able to self-sustain itself, but like, we don't know. So this level of interconnectedness maybe indicates that percula clownfish aren't, and this is me getting into extrapolating, so we don't know. But this might indicate that local adaptation for percula clownfish is not a very big concern, or it's not something that they're heavily selected for. So, and that would make some sense because anemones are pretty specific in their habitat preferences and needs. But I think that also means that we should be very cautious and maybe a little overzealous in how we protect these reefs. So Ocean Conservancy has been working with the Papua New Guinean government since the 90s. And so if you want to really look at cool projects, maybe check them out. You can look into the authors and try to figure out how you can contribute to their work. Whoa, continuity error. So I have a confession to make. I did most of the filming for this video about 5 weeks ago. And when I had gone to edit it, I was like, I don't think I closed my idea very well. And I thought, I'll just do some reshoots and it'll be fine. And then since then, I've traveled to Colorado Springs. I've moved. I'm actually in a new filming location in a new home. And then I gave a talk at the Missouri Aquarium Society in St. Louis, and I brought back about 30 fish. And I spent the last week making sure they were all situated before I had bandwidth to do anything else. And so here I am. So the point I really wanted to make with this is that I think most aquarists wouldn't even gotten to the point of trying to figure out a collection location for their fish. And I think that in and of itself is a disservice to breeders. But beyond that, I went down this rabbit hole of wanting to know more about the roots of the fish that I had. And it took me in directions that I didn't expect. And I had a lot of fun with it, and that's kind of the point of all of this. You're trying to have fun. And so I wanted to maybe share that a little bit with you guys and encourage you guys to go down your own rabbit holes. And please check out the literature in the description of this video. I would love to hear in the comments section rabbit holes you've gone down, and maybe give suggestions for rabbit holes you would like to see my perspective on in the comments section. But all that to say, I hope you guys stay safe, stay curious, and just have fun with it.